The overall goal of this procedure is to prepare and run 384-well assay plates for high-throughput analysis of a highly homologous group of genes, such as the type 1 and 3 interferon subtypes. This is accomplished by first preparing standard serial dilutions used for running the assay plates. Next, the primer probe and no-template control working stock mixes used for making the plates are prepared. Then, the 384-well assay plates are prepared using an automated multi-channel pipetter, and they are dried down for storage. Finally, the 384-well assay plates are run and analyzed. Ultimately, the high-throughput quantitative real-time RT-PCR assay is used to define interferon expression signatures in tissue culture models studying pathogens, or in clinical samples in the context of infectious or autoinflammatory diseases.
The main advantage of this technique over existing methods like quantitative RT-PCR using standard linear probes or cybergreen is that molecular beacon and log nucleic acid fluorescent probes can be used to discriminate single base pair differences between highly similar sequences.
After thawing, vortexing, and briefly centrifuging a standard and salmon sperm or ssDNA, prepare the ssDNA water mix for the 17 standard dilution sets by mixing 51 microliters of ssDNA with 20.3 milliliters of water. Dispense 190 microliters of the ssDNA water mix to the first tube of an 8-tube PCR strip and 180 microliters to the next 5 tubes. Perform a 1:20 dilution by transferring 10 microliters from the 50 picomolar standard stock to the tube with 190 microliters of